Okay, welcome everybody to the second event of Darwin Days 2023. My name is Warren Ullman. I'm the director of the Paleontological Research Institution and the co-sponsor of Darwin Week with Public Affairs at Cornell University, to whom we're very grateful. This year's theme is the Age of Fishes, which is in celebration of the Devonian, so we're talking not exclusively about early fish evolution, but mainly about early fish evolution this week. And so before I introduce our speaker tonight, let me mention that we have several other events scheduled for the rest of the week. Today is Wednesday, I think. On Thursday, we will have a panel discussion. We have three speakers talking about various aspects of early fish evolution and early tetrapod evolution. That's tomorrow night at 7 o'clock. And Friday night at Ithaca Bakery, a live event, we will have a trivia night. So many of you probably know that if Ithaca Bakery has great trivia nights, we will have our annual trivia fest on Darwin. So please come to that. On Saturday at Museum of the Earth, from 10 to 1, we will have a family day open house, all things Darwin and fish related. And then on Sunday, from I think 11 to 5, we will have also have a family day at Cayuga Nature Center. And we have lots of things Darwin and fish related at the Nature Center. So that's the rest of this week. Please come to as many events as you can. And more detail is available at priweb.org. So our speaker tonight, it's my great pleasure to introduce our speaker tonight is Willy Bemis. Willy is both a professor at Cornell in ecology and evolutionary biology, and he is the faculty curator of ichthyology at Cornell, which means he's in charge of the fish collection. Willy got his PhD at the University of California, Berkeley, a long time ago. And then he spent a long distinguished career at the University of Massachusetts at Amherst before coming to Cornell as the director of Scholl's Marine Lab. And he retired from that a few years ago and has had his third or fourth career returning to his love of teaching and doing research on fishes. And he is one of the world's most distinguished experts in the topic he is going to talk about tonight, which is coelacanths, lungfishes, and land vertebrates. Willy Bemis.
Thanks a whole lot. It's a great pleasure to be with all of you. I must say, I think the Darwin Days events really do a lot for the community as a whole. So I hope this is a terrific success. And you didn't say anything about the new lungfish exhibit. So I'm going to just say a couple things about this because it's really exciting to me. That's the long story. You want the long story? Sure, I'll tell you the long story. So about 2007 or 8, my colleague John Friel at the Museum of Vertebrates at Cornell adopted a lungfish from a pet store in Cortland. In those days, it was somewhat easier to get imported lungfishes. They're imported legally, but they're not very commonly imported. At any rate, he grew this lungfish up and she was a great success in his tanks. But along about 2014, getting ready to leave for a new job down in Alabama, John approached me and he said, Willy, I know you worked on lungfishes. Would you like my lungfish? And I said, no, John, my fish keeping days are over. I'm really not able to take care of fishes the way I used to. And about 6 weeks went by and he said, Willy, I'm getting ready to move. Are you sure you don't want the lungfish? And I said, I'm really sorry, John. I just can't. I don't have the time or the money to cope with a big aquarium setup. So I thought that was that. Well, our daughter was a Cornell undergraduate and she'd never let me visit her apartments or rooms. And I thought that was great. She needs her independence. I live in the same town. And she said to me one day in March of her senior year, Dad, would you like to see my apartment? And I said, sure, that'd be great. She says, good. Then you can see the lungfish. John had given her the lungfish. And of course, that meant that it wound up in my care when Kate went off to her next adventures. So Jane lived with us for many years, and I have finally convinced Kate that she needs to be at the Museum of the Earth. So there she is, installed in a brand new tank. I'm really super excited about this. She's a lovely animal. She likes people. She loves to be watched. So if you go to the museum, be sure to check out Jane, whose new name is, what is it?
Tembe.
Tembe?
Yep.
Tembe, which means?
To walk in
To walk
East African language.
in East African language. Another good name would have been Kamongo, which is what they call lungfish in Kenya, Kamongo. But that's a wonderful name, and I'm really glad you've recognized her for that. So hopefully I'll get a chance to see her, and she's going to quickly outgrow that tank warren if you keep the water warm. All right, well, tonight I'm going to talk about coelacanths, lungfishes, and land vertebrates. And this is actually a collection of organisms that have really translated to a lot of public interest in fish biology and evolution. And there's a whole series of cartoons about lungfishes and their allies. If I can get this to go ahead. This is a wonderful one by cartoonist Mick Stevens. He does did a series of these for the New Yorker. This one says, why don't you just stay and work on being a better fish? And that's a logical question to ask. Why did fishes end up evolving, some of them, into tetrapods? What was that all about? So we're going to explore some of these things tonight. Now, the group I'm going to talk about and focus on are the Sarcopterygians. These are the lobe-finned fishes, and you are a Sarcopterygian yourself. And here are the characters that let us know that. All of these animals from that node on down have a humerus and a femur. They have a single bone at the bases of their limbs. And that's the characteristic that makes them Sarcopterygians. The word Sarcopterygian refers to fleshy fin. And in addition to having a single bone, we also have lots of muscles associated with that bone. So that's the story of what Sarcopterygia means. And all of these animals are Sarcopterygians, from coelacanths and lungfishes to mammals. But I'm going to zero in to focus on tonight the biology of these two living groups, the coelacanths and lungfishes. And then we're going to talk a little bit, very briefly, about some of the topics that Ted Deschler talked about on Monday night regarding Tiktolic, this fantastically interesting fossil material that he and others recovered from the high Arctic. So there we are, Sarcopterygia. Now I have to start any talk that's going to talk about deep time with a quote from John McPhee, who's one of my favorite people. And we were fishing that day on the Delaware River long ago now. And he coined the concept of deep time in a book that he published in 1980. And then he said, numbers do not seem to work well with regard to deep time. Any number above a couple of thousand years, 50,000 years, 50 million years, will with nearly equal effect awe the imagination to the point of paralysis. So what I want to try to do for you is to teach you a little thought experiment in deep time. And it works like this. Say you're sitting on the banks of the Delaware River there, and you're listening to the birds, hear the wind through the trees, watching the fish, whatever happens to be that you're doing. And then what you need to do is you need to think about what that spot was like 10 years ago. Was it the same? Probably more or less. How about 100 years ago? Well, maybe not so much, because the people who lived around that river 100 years ago were different. Now cast back 1,000. Even harder to predict what that river would have been like 1,000 years ago. 10,000. What was going on with the Delaware River 10,000 years ago? Recovering from ice. 100,000. Anything you see or hear today going to be the same? Well, there'll be some similarities, but the farther you go back, 1 million, 10 million, 100 million, it gets harder and harder to predict what this spot on the planet would have looked like. And this is a really humbling experiment if you carry it to its logical extreme, because tonight we're going to be talking about things that happened 370 million years ago in order to understand Sarcopterygian biology. So, deep time is really the core of what I'm going to be speaking about, even though it goes unsaid most of the time. So here is a chart for deep time that features the Sarcopterygians. All the animals on this chart, all those little silhouettes, are Sarcopterygians, ranging from coelacanths up there at the top right to the lungfishes, the two families alive today, and to tetrapods down at the bottom, represented by salamanders. Now, there's a convention in this kind of tree, which is called a time tree, because it depicts time going across and phylogeny going with those branches. Now, the thin black lines in this tree basically indicate what we think we know about the evolutionary relationships among these organisms. And then there's some other details on this that I will get to in a second or two. But what I want to point out to you is that our time, the deep time we're going back to and celebrating this week for Darwin Days, is the Devonian. And if you look at this chart, you'll see that many of these fossils that we're looking at come from the Devonian, and that our group alive today, Coelacanths, is present there in the Devonian, and so are the lungfishes. So that's why they capture people's imaginations, because they go way back in time and help us get some kind of idea about what things in the Devonian might have been like. So I want to focus here on one thing, which is that we're going to zero in and look at some things that are right about there, about 370 million years ago, and in particular a locality up in Canada that I'll talk about a little bit. Okay, a couple other conventions for you. Well, before I get to those, I'm going to talk about this locality in Canada, I guess. So this is a really great place that I've never been. I have to actually make a pilgrimage to Miguasha, Quebec. It's a national park in Canada. It's a UNESCO World Site. And why is that so? Because it preserves a slice of the Devonian in exquisite detail. There's a shot from the water looking up at the cliff, and out of those cliffs come things like this animal here, the Devonian tetrapodomers, Houston, Operon, Fordi, that I'll talk about just a very little bit as we get to the end of the talk. But let's put that site at Miguasha in some context for you. Okay, this locality captures a slice in time, about 370 million years ago, and it actually extends to either side of that. So it's more than just, a short slice. It's a big slice. It's an example of a fossil Lagerstätte with exceptional preservation of fossils. And because they're so well preserved, they've had a disproportionate impact on what we think we know about Devonian animals. This is often the case. We zero in on the best preserved material, and it gives us insights and ideas, but there's a lot of stuff that we don't discover. These just happen to be the ones that are well preserved and that we've discovered. Now, the exceptional preservation of the fossils in this site is attributed to a lot of things, but in particular, this was probably an estuarine environment at the time the fossils formed, and there was very low oxygen in the water. So when the fish died, they didn't decay on the bottom of the estuary. And because it was an estuary, there was a large amount of sediment that covered them over. So it's a really exciting place to think about in the history of vertebrates. Now, as I said, we interpret it as an estuarine habitat, and this is a wonderful painting that shows the fishes living here in Miguasha. And when I say estuary, we're not talking like a little tiny estuary. We're talking about an estuary the size of the Mississippi, where it comes out into the Gulf of Mexico. A huge, huge place. And why was that happening? Because there was a spine of gigantic mountains running just to the west of this locality, and all that sediment was washing out into this estuary. Okay, now I like to show this particular chart. This is one of those amazing paleo maps by our friend in Colorado. What's the name again? Should put you on the spot.
Ron Blakey.
Yeah, Ron Blakey. Thank you. I actually paid for this one because they don't like you to use their maps. But I really appreciate the work that he does. And I like this because it shows Ithaca. That's supposed to be there. Did it not come out? There we are. There's Ithaca on the west side of this spine of mountains. There's Miguasha. And then there are a couple other localities up there. And I don't know if Ted talked about East Greenland last time, but his Tiktolic locality is up there. And you can see that at this point in time, about roughly 370 million years ago, the equator passed through North America at that kind of angle. So at the time that Tiktolic was alive, it was relatively near the equator. The East Greenland locality is another very important place because that's where we have the earliest tetrapods, Ichthyostega and Acanthostega, that come from that spot. We'll talk a little bit about them, but not much today. Here's Miguasha, and you'll see why I like it as an example. It has all the fishes. We have coelacanths in the form of an animal called Migushia, and we have Paralipiforms, Holoptychius. I'll tell you a little about those guys. Scamanacea is an example of a lungfish that comes from there. Operon, that very famous fish that I showed you in the previous slide. And Paralipus, which is a very early actinopterygian fish. The actinopterygian fishes today, about 35,000 living species of actinopterygians. And that radiation is one of the most intense success stories in evolution, but it's basically much, much later in time than the Devonian. So there were a few of these actinopterygians running around then, a lot more today. Okay, I think that probably is good for that slide. So let's speed ahead here and talk a little bit about Sarcopterygians and some more of these conventions. One of the conventions is that the thick blue lines on my chart indicate the occurrence of fossils. So here's what's cool about coelacanths, for example. We have coelacanth fossils from throughout the Devonian, and there's quite a bit of diversity. They don't all look alike. There's a lot of anatomical diversity in the coelacanths from the Devonian. And after the Devonian, they kind of stabilized on a body plan, which is pretty much consistent from, say, the Carboniferous to today. One of the most famous things about coelacanths as a lineage is that they have this huge gap extending from the end of the Cretaceous all the way up to the recent, all the way up to the coelacanths that we have alive in the world today. And it was the occurrence of that gap that convinced everybody prior to 1938 that these animals were all extinct. So it's a famous, famous story that makes them celebrated and iconic. Okay. Now, I have one other convention I want to point out here. It's just basically how the splits work on this thing. These splits are estimates, right? The splits between this animal up there, the Onychodontiform, and the coelacanths. We don't really know exactly when that happened, but we think it must have happened in the Silurian based on the oldest fossils of the two groups that are included in Actinistia. So Actinistia includes two lineages that I'm going to talk about tonight. One are the Onychodontiforms, and the reason I'm going to talk about them is that we have them in the Devonian of New York State. And one of my happy experiences last spring with Warren's student Will Hooker, who was also a student of mine, was working on Onychodont teeth from up near Syracuse. So it's a cool local thing, Onychodontiforms. And then the other line, of course, is the one that leads to coelacanths. Let's look at Onychodonts for just a second here. They're commonly referred to as the dagger-toothed fishes because they have these spiky, spiky teeth. This is actually the specimen on display over at the Museum of the Earth. And sadly, here in New York State, we mostly recover scrappy fossils like this. But if you work in the Australian Devonian, you find more complete Onychodonts. So here's an example of one of those guys. It's been elegantly drawn by our colleague, Brian Chu. And what they have is they have this tooth whorl. That's what that is. It's right out there at the tip of the lower jaw. And it's been speculated that these animals had a biology that was similar to a moray eel on a reef. They lived in holes. They came charging out and slashed things and brought them back in to eat. I wish we had Onychodonts alive today. I think they would be really fascinating. But sadly, they disappeared at the end of the Devonian. So let's press ahead and talk about the coelacanths. The first point you need to remember about coelacanths is that there are two living species of coelacanths, Latimer columna, which was first caught in 1938 and described in 1939, and Latimer monatoensis, which was described much later in the 1990s. The interesting thing about these two species is that they occur on opposite sides of the Indian Ocean. So the original coelacanth discovery was off the east coast of South Africa. And the subsequent discoveries up to the 1990s were all from areas either adjacent to or islands offshore from the East African coast. And then this wonderful other species showed up in a market in Indonesia. And it turned out, based on genetic and morphometric data, to be a separate species from the East African coelacanth. So there are two of them in our order Coelacanthiformes today. And what I want to point out to you are some of the basic features that make coelacanths such interesting fishes. This is a classic figure that we redrew from the studies made in France by Milot and Anthony in the 1950s. They published beautiful, beautiful anatomical books. Some people said it was the end of comparative anatomy because they did such beautiful books. Of course, that's not true. comparative anatomy is alive and well, probably better than ever at this moment in time because we have so many new techniques. But nonetheless, they illustrated their stuff beautifully. So we redrew this from their work and from others. And I want to point out, we're just going to go around. I always tell my students, when you're looking at fishes, you start with the fins. So it's convenient to start with the first dorsal fin. If you look at that fin, it looks like the fin of maybe a perch, right? It could be spread and collapsed a little bit. It has spines that hold it up. Nothing too special to write about there. But look at the second dorsal fin. It's not like the first dorsal fin at all. It has a muscular base, and it has the ability to move around as the fish is using it to swim. And it's essentially on a stalk with a single bone at its base. If you look at the caudal fin of this animal, you can see it has three lobes: an upper lobe, a middle lobe known as a tuft, and a lower lobe. And we call this type of fin a difficircle caudal fin because the skeleton that supports it is absolutely symmetrical. It doesn't turn up. That's a big deal in fish systematics if you have a turned up tail or a straight one. So that's why I put the word difficircle up there for you. Here's the anal fin. Like most fishes, the anal fin and the second dorsal fin are very similar to each other in terms of their morphology. And that's probably because they're regulated by the same Hox gene complexes expressed along the body axis of the fish. Here you can see the muscular base of the pelvic fin shown there. And here is the muscular base, the Sarcopterygian feature of the pectoral fin. And so the fins are really interesting, and they tell you a lot just by that. Looking at the head, there are a couple of interesting things there. One is they have an incurrent nostril and an excurrent nostril on each side of the head. So the nostrils are collecting water, pumping it through the olfactory system, and then out on the side of the head. That's a thing that's different between you and coelacanths. Our nostrils open inside our oral cavity, at the back of our oral cavity. And that's something that coelacanths don't have. You can also see three little openings on each side. That's for the rostral organ, an organ that we interpret as an electrosensory organ, similar to the electrosensory organs of sharks. But coelacanth anatomy is absolutely fascinating. I spent years and years and years thinking about it. Okay. Now, let's talk about some fossil coelacanths for a minute. I mentioned that there are coelacanths at Miguasha, and the genus Miguashia is an example of coelacanths from that place 370 million years ago. And if you compare this with what we just looked at, you'll see several things. You'll see that the fins look moderately similar, except for the caudal fin. It's got this strong upturned lobe, and that upper lobe has beneath it a bit of a fin. This is the condition that jawed vertebrates inherited when they developed jaws. That's basically an old thing to have your tail curled up like that. And it was present in the Devonian coelacanths, but lost in the Carboniferous and post-Carboniferous coelacanths. The other thing that people don't often appreciate about coelacanths is how big they got. This is Mawsonia, which is obviously a very gigantic coelacanth in comparison to that sketch of a person there. And in this case, it has that triple tail with a tuft, just like the living Latimeria. And that body plan was pretty much stabilized for millions and millions and millions of years. Did they all live in marine environments? No, they lived in freshwater environments. They lived in marine environments. They were all over the world. And we find them at low diversity. They're never like, hundreds of species of coelacanths. There's usually one or two at localities stretched from the Devonian to the recent. So they're a really fascinating group, and I love them. So we'll talk about them a little bit further. One of the things I like to emphasize about coelacanths is their role as iconic species. And what do I mean by iconic? It means that they have an impact outside of their biological impact. Like we talk about politicians as being coelacanths if they're out of date. Or we have one of my favorites, we have breweries named after coelacanths. This is the Coelacanth Brewing Company in Norfolk, Virginia. And I was there like a week before it closed because I had to go there. I couldn't not go to this place. They had a very famous statement about their place. They said, no yoga, no trivia, no pumpkin beer. And I loved it. It was a wonderful evening there. We bought a keg and a bunch of glasses to celebrate my daughter's defense. So she took me there for this event that night, and they were closed 2 weeks later. It was really sad, really tragic. I don't think we're going to even get them to reopen or make anything, because we talked to the owner and he's like, yeah, I'm going to do something else now. So, you know, things come and go. Okay, and I think the other thing that's really funny about coelacanths is that they really are, people make pilgrimages to see coelacanths. This is the original coelacanth that was described by Marjorie Courtney Latimer. She didn't describe it, she found it, and Smith described it. I always ask people, oh, I do. Thank you for pointing that out. I was so jazzed about talking, I didn't even plug it in. There we go. That good? No problem. Always prepared. So people make pilgrimages to go and see Marjorie's coelacanth there in South Africa, in East London, South Africa. So one of my graduate students, Heath Cook, who's actually doing research in South Africa this week, made a pilgrimage for me to go and photograph Marjorie's original coelacanth. So they really are iconic animals. Now, I have some reasons for that. I have a few, and I put the whole Indian Ocean here because I often ask people what they think the future of coelacanths is. I could solicit some ideas, or we could do it in the chat. But I'll tell you, I think the future is really interesting. And that's because we found them here, and here, and here, and here, and all the way up the coast in Malindi in Kenya, a place that I've camped on the beach at Malindi, and I didn't have any idea there were coelacanths offshore. There's a new spot off Tanzania where they come from. They're coming from other places farther and farther afield. And I've encouraged my younger colleagues to get a boat and go out and fish all these spots, because I'm sure they're way more well distributed than we think they are. But the iconic things include, they have this great long fossil record that's inspiring, and you can see some really interesting things fairly easily. They have this cool structure and biology, living exclusively in marine environments today, but everywhere in the past. The stories about their discoveries are truly romantic, and there are many popular books that I love that describe how coelacanths were discovered, who did it, when, what the impact of that was. There's a lot of good stories about coelacanth discovery. And I think the last one is that they really do have a place, an established place in popular culture. Fascinating organisms. Okay, so now let's turn to lungfishes and their allies in a group that's called Dipnomorpha. Dipnoan means lungfish, and dipnomorphs are things that are like lungfishes and lungfishes. So this is this group that we're going to talk about first. These are Paralipiforms, which, like Onychodontiforms, have a terrific Devonian record, including lots of things from North America, but sadly are no more. They're called Paralipiforms because they have big holes in their scales, so they're quite distinctive in the record when you find them. And the purpose of those holes has been debated for a very long time. I'm not sure I agree with any of the interpretations. They have holes in their scales. That's the fact. And then the lungfishes themselves, well, let me show you Paralipiform, and then we'll talk about the lungfishes. This is what a Paralipiform reconstruction looks like. Paralipus, that's quite an imaginative reconstruction, because we really don't have anything like a whole body of Paralipus. It's quite a bit of artist license there by Dimitri, who is a wonderful paleo artist, by the way. And I mentioned the large pores. These are the scales, and you can see those holes in them. Those are the large pores that they have. Pore-bearing scales is what that means, Paralipiforms. One of the most interesting Paralipiforms, a little bit later than Paralipus itself, is this animal, Holoptychius, which comes from Miguasha. And I have a cast of this specimen, which was at Dartmouth, but now is at the AMNH. The cast, I think I need to give to you, Warren, because it's a wonderful cast. And it's a relatively complete body. You have portions of the head are missing, but you have the whole trunk and all of the fins. It's a really, really beautiful fossil. Sadly, Paralipiforms are no more. Now, I want to turn to lungfishes here. And here are the lungfishes. And you'll see again that this is a clade that has beautiful record in the Devonian and the Carboniferous. And the two extant families, Neoceratodontidae for the Australian lungfish and Lepidosarin for the African and South American lungfishes, they have a great fossil record from the Cretaceous to recent. And there are other members of the lungfish tree that I haven't shown on here just for simplicity. There are about 60 or 70 genera of fossil lungfishes, and many of them are very well preserved. They're very well studied. And to my mind, you know, I never understood why coelacanths got all the glory, because I think lungfishes are equally interesting. In fact, in some ways more so, because the biology of the living species, which I'll talk about a little bit, is actually very, very fascinating. Okay, so we've got these two groups of lungfishes alive today. And I want to talk first about some Devonian examples, and then we'll press ahead and deal with our living taxa. Now, I love this particular specimen. This is an animal called Chirodipterus. It comes from the Gogo Formation in Western Australia. And the Gogo Formation is a wonderful spot that I also have never managed to get to, but it is admittedly out in the middle of nowhere. And essentially what you're looking at is you're looking at a Devonian reef face, and the fossils are in nodules off the edge of that reef face. The nodules are limestone. And what that means is that they could be prepared using the acid transfer method. The acid transfer method was developed in the 1950s at the museum in London for use on fossils like these wonderful things from Gogo. And it's really sort of a magic method. If you have a fossil in limestone and you can expose part of it, then you embed that part in a very thick polyester resin typically, and turn the thing over. And then you put it into a bath of dilute acetic or other organic acids. And you go back every day, wash off the stuff that's dissolved away, stabilize the bone with fingernail polish, basically, and then you put it back in the vat. And by doing that, you can essentially retrieve a three-dimensional skeleton of the animal as it looked in life. And that method applied to the study of these Gogo lungfishes was happening about the time I was getting interested in fossil fishes. There's a very famous paleontologist in London, Roger Miles. He wrote a whole book about his lungfish experiences with these Gogo Formation fishes. And it was really the first time that I realized just how extensive the fossil record for these lungfishes is, because there's not just one genus from Gogo. They're not two, they're like 4 or 5 genera from Gogo. And the Devonian of Australia is loaded with lungfishes from other places too. So there's a lot of diversity. And you can probably appreciate that that fish is quite a bit different from what we have alive today. One of the things that it has is a very solid skull roof. It also has better ossification of the bones. And although I didn't show it in that one, you can see it in this cartoon at the top here. These fishes retained that upturned or heterocircle tail that was lost in coelacanths on the line that leads to today, and it was lost in the lungfishes on the line that leads to today. You can see that the lungfishes have this kind of feathery shaped fin. Now, when you go to look at our lungfish at the Museum of the Earth, you're going to notice that her fins are nothing like the fins of a Devonian lungfish or even of the Australian lungfish today. They're much, much more reduced. But this is a really important fin in terms of interpreting how land vertebrates got their limbs. One of the things that lungfish is pretty much stabilized on by the end of the Devonian was a pattern of tooth plates. And these are tooth plates that have a radiating pattern like this, and they're used by lungfishes today to crush up plant material, snails, or in the case of African and South American lungfishes, lots of fishes. The plates are quite different in the South American and African lungfishes from what we see in Australia, and that was a big part of the dissertation work that I did to understand how those tooth plates form, develop, and are used. Okay, so those are some Devonian lungfishes. Let's sprint ahead here and talk about are living forms in the context of their fossil history. And one of the most interesting things about lungfishes is their biogeography. So these things were there before Pangea got assembled. They were there after Pangea split up. And amazingly enough, in the Mesozoic, they were virtually everywhere on the planet. Find fossil lungfishes in the Western United States, you find them in Belgium, you find them in Africa, they're all over the place. And I've cartooned for you the Triassic, Cretaceous, and Cenozoic fossil localities and the extant Neoceratus locality there in Queensland. All those purple and green things, long gone. Lungfishes today only occur on basically southern continents. And for a long time, people said, oh, yeah, it's just a Gondwana distribution. And then this wonderful colleague named Keast in 1977 generated the original versions of these maps and said, no. They went extinct everywhere except South America, Africa, and Australia. Now, I've got a little tree here just to remind you that the Australian lungfish, Neoceratus, is quite different from Lepidosarin, the South American lungfish, and from Protopterus. And the fins are just one place to start looking for those differences. You can see they're thread-like in Lepidosarin and Protopterus compared to the wide paired fins that we see in Neoceratus. Now, everyone loves Australian lungfishes. And one of the things that makes the comparisons with coelacanths so fun is that lungfish fossils were found before Neoceratus was found. The first lungfish fossils were found and described in the 1820s. Coelacanth, I would say our Australian lungfish wasn't found and described until the 1870s. So 50 years went by. And that's somewhat parallel in the 19th century for what happened with coelacanths in the 20th century. But the popular press didn't grab it the way they did in the 20th century for coelacanths. They're absolutely wonderful and charming animals. They can live for a long time, 80 years in the case of an animal at the Shed Aquarium, an animal called Grandad. I knew Grandad. He was a wonderful fish. They have now a very old one, now the oldest in captivity, we think, at the California Academy of Sciences Aquarium in San Francisco. I went to Australia in the late 70s, early 80s, and I was studying larval Australian lungfishes. And we were also collecting lungfish for my colleague, Glenn Northcutt, who was studying brain anatomy of lungfishes. They live in these unbelievably great places. Like this is the Brisbane River, which literally does flow through downtown Brisbane, but you have to go upstream, not very far, and it looks like this. And at night, if you go out at night with a boat and illumination, you'll see those lungfishes just kind of lying like logs along the bottom at the sides of the river. What I love about this is it's a 2019 or 2021 photo. This guy is using a drone to monitor lungfishes in the Brisbane River. So I just think that's wonderful. Okay, some fun facts about these guys. They don't depend on air for their respiration. They can breathe air, they have a lung, but they rely mostly on aquatic ventilation of their gills. And they are slightly carnivorous, but they mostly eat plant material. Another thing that's very interesting is they don't show any parental care at all. And that's a big contrast to what we see in both the South American and African forms. So here's an African lungfish, Protopterus. This is what our friend at the Museum of the Earth is. And, superficially, it looks kind of eel-like, but these things are not eel-like in their behavior. Imagine a tube of muscle the size of your arm. They are very powerful. They don't move very much, but when they want to move, they can move as much as they want, as fast as they want. When I was in East Africa, collecting lungfishes for my work, one of the things that was a very, very funny story, I'd go every morning to the landing place where the fishermen were bringing in lungfishes and everything else. And I would barter with them to buy some lungfish for my work, whatever. I got there one morning and they said, "Oh, you just missed the big lungfish." I said, "Well, where is it?" "It's on that bus. The fishmonger bought it and she's taking it to the market." So I followed the bus to the market. And I got to the market and I found the woman with the lungfish. It was 1.5 meters long, a big lungfish. And I wanted to buy just part of it because I knew she would charge me whatever she could get for the whole thing. I said, no, I just want the head. And so she's putting her knife down on this lungfish and warning me. She says, do not get your hand near that fish. It will bite it off. And they really are incredibly powerful. If you feed Jane and you watch Jane feeding, it's pretty interesting. They can bite a broomstick in half if they want to. So fascinating, fascinating animals. That's the little tiny opercular opening that I've indicated there. And right behind the opercular opening in young African lungfishes, you sometimes have the obvious remnants of their external gills, which they have early in development, and they retain them to varying extent as they grow to adulthood. Typically, they're not very important in respiration as adults. There's some really unusual things about African lungfishes. They estivate, which is summer sleep. So when the pond dries up, they dig a burrow like that, curl up in that burrow with their head just at the water level. And if everything dries out, they just form a mucus cocoon around themselves and stay that way until the water returns. Now, the fascinating thing about that is it actually made it possible for physiologists to take lungfishes from Kenya to New York in the 1930s. What they did was they would take a can, coffee can size can, put a lungfish in it with mud, and then let the water evaporate. And then they just shipped the cans home and rehydrated them at the New York Aquarium. So that's kind of a fun story. That guy wrote a great book, by the way. The book is called Kamongo. And the subtitle is The Lungfish and the Padre, because he was traveling across by boat through the Red Sea and along East Africa and having conversations with this padre along the way about lungfishes and the meaning of life. It was a wonderful book. Okay, this is a wonderful nest picture. Lungfishes make nests. These African lungfishes build a nest and they guard their young in that nest. Very, very different from what we see in the Australian form. Now, if you watch the lungfish at the Museum of the Earth, you'll probably, if you stick around long enough, watch her air breathe. And this is very interesting because it's quite different from our air breathing. Lungfishes practice what's called buccal pumping. This is the buccal cavity, your oral cavity. And they expand the floor of the oral cavity to suck in air from the outside. Then they open the glottis. The lung has elastic fibers in its wall. The air goes out into the glottis, and they pump and mix the air with the fresh air in there. Then they close the mouth, raise the floor of the mouth, and pump the air into the lung. So the lung is pressurized. The glottis is all closed, and then they sink back down to the bottom. And they'll repeat this. It used to be one of my favorite assignments for a new student in my lab. I'd say, watch that lungfish, see how often it breathes. And if they lasted an hour and got it to breathe, then I knew that they were really interested in lungfishes, right? Okay, so that's that animal. Now, South American lungfishes are also very interesting. One of the coolest things about these guys, this was the first species of living lungfish to be described. It was described by a guy named Fitzinger in 1837. And he called it Lepidosarin, lepido meaning scaly, because it has scales, and siren, because to him it looked like an amphibian, looked like the siren, the amphibian siren. And that set off this long debate. And in many ways, that debate of whether lungfishes were fishes or land vertebrates or something else, that debate really didn't get settled until during my lifetime. And it was finally settled in the 1990s by my friend Axel Myers, who showed, using molecular phylogenetic methods, that there was no way that coelacanths were more closely related to land vertebrates than the lungfishes were. But there's an interesting period in the history of this where people were so discouraged about finding out what the immediate outgroup for land vertebrates was, that they put lungfishes off in other branches entirely. Like there was a school in Stockholm that suggested for a while that lungfishes were actually more closely related to sharks. They just didn't want to have them anywhere near the origin of tetrapods, but they are very close to the origin of tetrapods. Once they were described by Fitzinger, his locality was right in the middle of the Amazon basin. And that locality, nobody went back there, and they couldn't find any Lepidosarin in the middle of the Amazon for a long time. They were then rediscovered near the mouth of the Amazon and all the way up in Peru before they, again, were discovered in the middle of the Amazon basin. And this is a very good locality. I visited this place with Christina Cox Fernandez, who's just one of the most wonderful South American ichthyologists you could possibly imagine. She did her PhD work in Manaus, and this is very near Manaus. It's where the Solomos and the Rio Negro, the two rivers that form the Amazon, meet at the meeting of the waters. And this little island is out there near that meeting point, and that's where they collect lungfishes for the Manaus collections. Now, like African lungfishes, these guys also guard their young in nests, but they do it with an even more intriguing way. So males are doing the nest guarding, and they develop these structures on their pelvic fins. They're called pelvic gills. And so they're sitting there taking air in at the surface, and these pelvic gills are just like little threads that are setting out into the nest. They can lose oxygen from the pelvic gills into the water where the nestlings are. So I think that's just really a great story. Somebody really needs to follow this up with field work, because there's never been very convincing field work to back up the story based on the experimental induction of these pelvic gills. You can induce them by injecting testosterone, but it's a wonderful story, and somebody needs to go back and explore it in the field. Now, I just have a few more things I want to point out for you. I want to talk a little bit about tetrapodomers, even though this is properly Ted's area, and I don't want to repeat anything that he talked about the other night. But I do want to point out that there are a whole variety of fascinating Devonian tetrapodomers, ranging from rhizodonts, which are giant fishes. We have them from the Carboniferous of Pennsylvania, 6 or 8 feet long, even bigger in some cases. Osteolepis, very interesting fish from Scotland. Eusthenopteron, not only in Maguasha, but also in Nevada and also in Australia. Panderichthes, there's Tiktolic. Acanthostega ichthyostata, these early these earliest tetrapods, really. And then a series of other tetrapods leading forward till we get to the crown tetrapod group that includes us and all of the other tetrapods alive today. So what is it about these guys that I like particularly? Well, here's Eusthenopteron. And I want to tell you a story about Eusthenopteron because this for many years was the best known fossil fish anywhere in the world. Why? Because there was a scientist in Stockholm, Erik Järvik, who made it his life's work to understand the anatomy of this fish. And in those days, they did not have CT scanning. So what did you do in order to study the internal anatomy? Well, it sounds really crazy, but they'd grind the fossil up. They grind it up. Not this one. This one you can see at Cleveland. It's in the Cleveland Museum of Natural History on exhibit. But so I have a head. I've forgotten the number. It's P something or other, 47 or so. I can't remember it, so I don't know. But it's a very, very famous fossil, even though it no longer exists. They would grind a section and then take a photograph and a peel of the surface. Grind another bit, take another photograph and a peel. And using that serial reconstruction method, Järvik interpreted the anatomy of Eusthenopteron in incredible detail. Did he get everything right? No. Was he wrong about a lot of things? Yeah, he thought that tetrapods had 2 separate origins, that frogs and mammals came from one group and salamanders came from another group of fishes. That diphyletic theory of tetrapod origins has been off the table for decades at this point. But he was an extraordinarily gifted anatomist, and he applied those skills to the study of Eusthenopteron. So I love the story of Eusthenopteron, if only for Järvik's wonderful work. Here is Ted's animal. This is Tiktolic Rose. And I put it up here partly to remind you that you can see this cast at the Museum of the Earth, and it's worth making a special trip just to see this animal. Why? Because you can see that it has a neck. The back of the skull is not connected to the shoulder girdle. And that's the major cool thing that made Tiktolic so special when I first heard about it, certainly. And I knew it had been found because I was hanging out at Harvard for some reason a lot in those days, and they were talking about it. And it was years and years and years before it was published, but it was a really, really exciting fossil for so many reasons. So I know Ted probably gave you guys a great introduction to all of this. Okay, so Well, from that, what are the things that really separate our Devonian tetrapodomorph fishes from Devonian tetrapods? What are the big things? Well, here's a fish, and that's based on Eusthenopteron. And what I want you to take a look at here is that the pectoral girdle there in blue is attached to the back of the cranial skeleton. So it'd be like having your shoulder attached to the back of your head. You don't have a neck. And that bears a pectoral fin. And then you look at the pelvic girdle, it's not attached to the vertebral column at all. It's floating in space. And there's a pelvic fin that it supports. And the vertebral column is there, and it's nowhere near as sturdy a vertebral column as you need for being a land vertebrate. So if we go to the next cartoon here, here's based on a Devonian tetrapod like Ichthyostega. And the things you want to note are that they have a neck, and that the pectoral girdle is not attached to the cranial skeleton. That means you can swing your forelimb in order to move around on land. But the consequence of being on land is that you need some way to support the weight of the body. So one of the big changes is that you find the pelvic girdle now attached to the vertebral column. And so you disconnect the pectoral girdle from the axial skeleton, and you connect the pelvic girdle to the axial skeleton. And that pattern is basically observed across the rest of the land vertebrates with minor variations, loss of limbs, for example. Now, also important, if you're thinking about the transition to a land vertebrate, you have to think about how the body, the weight of the body cavity is going to be carried. And this involves major inventions in how you put your vertebrae together. You look at something like a Eusthenopteron vertebra there, that's straight from Yarvik, and you can see that, yeah, there's a centrum, that's the vertebral body, and it's made up of two parts, an intercentrum and a pluricentrum, and there's a rib, but it's not well ossified, and there's a lot of open space, it turns out, in those vertebrae. They're occupied by cartilage in life. In contrast, if you look at Ichthyostega, the vertebrae have become much more consolidated. The ribs are now much more important, and they're supported in two different places on the individual vertebra. So there are lots of cool changes, not just in the fins and limbs, but also in the types of vertebrae that we associate with land vertebrates. Now, I want to end this by talking about some work that was published just 2 years ago, again, out of Axel Myers Lab. You never want to underestimate Axel Myers' resources. This guy is a genius, a wonderful, wonderful colleague. And what he did was something that we all wanted to know about when we were in graduate school together. And that is, how can we tie the evolution of the pectoral appendage to its development? And here you can see we've got Latimer up at the top with a red line marking the axis that goes through the fin. There's Neoceratodus, the Australian lungfish. There's Eusthenopteron, Panderichthys, Tiktolic, and Acanthostega, one of these early Devonian tetrapods from East Greenland. And you can see that I've cartooned on these guys the arch as it bends during this transition. So here's Acanthostega. The axis goes like this through the wrist and fingers. So how did that relate to its development? Here's what Axel's team found. They looked at the expression patterns of a couple of genes that are involved in specifying the axes of limbs. They looked at HOXD13 and ALX4. And unusually, they were able to work on Australian lungfishes. They're captively raised now, and so there are people who have the ability to study early embryos and larvae. But what's going on here is that they showed that the HOXD13 expression was all along the post-axial side of the fin, and ALX4 all along the preaxial side of the fin. So there's very clear specification of posterior and anterior in that fin. Then they looked at gene expression during forelimb development of a mouse, because that's another great experimental animal. And here's what it shows. HOXD13 is expressed there and ALX4 there in an early stage. You get an intermediate stage where the Hox D13 expression is expanded a bit. And finally, you get to a later stage where Hox D13 is expressed in the hand and ALX4 is not expressed in the hand. And what this means is it's those elements that I've colored purple up there, purple down here, that's where the digits came from the backside of the fin of Devonian fishes. And there have been arguments about this for the last century. So this was a really important paper because it set those problems and questions to rest. Now, I have so many things I'd love to tell you about, but I'm going to stop now. And I'm stopping with another one of Mick Stevens' wonderful cartoons in which the fish are saying, I'm liking it already. So I'm happy to answer any questions people may have.
So I think people can hear me online. And if you would like to ask any questions, feel free to ask in the question and answer box. You can also raise hands, but I think it might be simpler just to have you type your question into the box. We have one already from Linda Ivany.
1 second, let me see if I can turn my volume off here so we won't have any reverb.
We have it. So we have two questions now, one from Linda Ivany and one from Michelle Song. I think maybe you can see these in your Q&A.
I should be able to.
If you take a look, I'm happy to read them, but you might, maybe it's easier for you to do that yourself.
Good one. Are others having issues with sound quality? I assume we solved that.
I think that was early on.
Okay, good. I don't know, handful, probably about 10 or so. Can't modulate.
I think those were all before.
Okay, good.
Started.
So the question came up about the second dorsal and anal fins are not paired. True. Why do they have a bone supporting them? Well, you need a bone in order to attach muscles that are going to move those fins around. So to the extent you're going to have mobile fins, you have to have some sort of skeletal support. It doesn't necessarily have to be well ossified bone. It could be cartilage, but there has to be a structure there that can support it. And it is not a neural spine. It's a separate ossification. And it's different from the paired fins. Now, one of the things that developmental genetics is showing us is that many fin structures have similarities in different fins. And it may be that there are essentially toolkits for making a fin. And there's a lot that's going on in that area that's beyond my expertise, really. But no, these are not equivalent to paired fins, though they may use some of the same toolkit genes in order to make them. Okay, so we've got another one. South American and African lungfishes, any theory as to why they evolved their elongated thin fins? Adaptation for navigating muddy habitat, that is a wonderful question. It's so difficult to construct meaningful tests of that type of hypothesis. And the reason it's difficult is that we don't, African and South American lungfishes are so different from Neoceratus. They've been separated probably since the Carboniferous. So that's millions, tens, hundreds of millions of years that they've been evolving independently. And I have to say, one of the most fascinating things about those paired fins of the South American and African lungfishes is their amazing regenerative capability. I had a lungfish I brought back from Africa. I called her Stumpy because she'd lost her fins during the travel. She was a wonderful fish. She grew them right back. And that ability to regenerate is one of the things that has attracted a lot of attention to lungfishes because they really do an amazing job. I don't think we could conclude that these are an adaptation for navigating in muddy habitats necessarily, but there are some interesting things that they do with them. I have a good colleague at the University of Chicago who showed quite convincingly that they put their fins in a diagonal gait arrangement, just like a salamander would. So they're walking on the bottom, even with those little thread-like fins. So it's not particularly, I don't think of it as an adaptation for moving through weeds necessarily. They're there, but they have retained that diagonal gait pattern.
Another thing I'd point out about them is that lungfishes in captivity use their paired fins, particularly the pectoral fins, to touch food. They find food with their fins. So there's a lot of different functions that they may be used for. And that wouldn't be something you could easily do in the Ceratodus. So they may have a more sensory function than a locomotor function. Can the relationships of lungfish? Yes, this is very interesting, actually, a great question. Can lungfish biogeography tell us anything about the separation of the continents? And here I'm going to focus on South American and African lungfishes. This distribution probably relates in some way to the separation of South America and Africa in the Cretaceous. And what's interesting about the South American and African lungfishes is that they do kind of look alike. And what's really fascinating is there are 4 species of lungfishes in Africa. And one of them, Protopterus doloi, which comes from the Congo basin, is very elongate compared to the others. And that's the area that potentially would have been in closest connection to South America. So there's some articles that talk about this similarity out there. I've never really speculated on it in writing, I don't think, but it's always struck me that Protopterus doloi looks a lot like Lepidosarin. Okay. Digits on pelvic appendages. This is a great question. Is the evolution of digits on the pelvic appendages similar to that for the pectoral? Broadly, yes, but in fact, we have much less evidence for pectoral fin anatomy in most of the fossil material we have from the Devonian. And that's because the pelvic fins were small. They're much smaller than the pectoral fins. If you look at something like Neoceratodus, yes, they have similarly designed paired fins in both the pectoral and pelvic limbs. You know, it's really great speculation for me to go beyond that to talk about how the pelvic fin evolved for land locomotion. Okay. I think caused the absence of, oh, this is such a great question. The absence of fossil lungfishes and coelacanths, unknown. It's a great question, absolutely great question. And these are the kinds of questions that motivate people to go out and look for fossils. So my answer would be similar to an answer I gave in a lecture this morning. Best way to figure that out is to figure out where that rock is going to outcrop somewhere on the planet and do what Ted did, go there. And if you let the rocks be your guide, you have a much more realistic chance of finding the material that you want. Now, there's no guarantee, but that's one of the exciting things about field paleontology. Would you agree with that? Yeah. Okay. So yeah, there's no evidence. This question asks, understanding the evolutionary history of lungfish to other vertebrates, some studies suggest that lungfishes are closely related to tetrapods, while others suggest they're more closely related to other groups of fishes. This is pretty well nailed down now by Axel's wonderful work in the 1990s. Prior to that, another really important paper was published in 1982 by a group of paleontologists and neontologists at the American Museum and the London Museum. That paper has the wonderful title of Paleontology, Plesiomorphy, and the Origin of Tetrapods, or something like that. It's a wonderful paper. I have a funny story about that. I had a draft, I had a manuscript of that paper that I'd somehow acquired. I think Don Rosen sent it to me. I was a graduate student in Berkeley, California. I didn't know anybody. And by happenstance, Gareth Nelson, who was a very important curator at the museum in New York at the time, was spending 6 months a year in Berkeley because that's where his wife was. So I asked her one day, I said, do you think I could go visit Gareth in New York? And she said, oh, he'd love to have you. So I talked my way into visiting Gareth in New York, and we had the most amazing week. I was basically trained as a pattern cladist by the end of the week.
But the most fun part of this was sitting across the desk from Don Rose and arguing every character in that wonderful paper. And I would say, I don't see this. And he'd say, you're not looking carefully enough. It's over there. And we had fossils and cleared and stained materials. It was just an amazing, amazing experience all around. So I would say, in answer to the question, there's really not much evidence that lungfishes aren't the sister group of tetrapods, and that coelacanths are outside the group that includes lungfishes and tetrapods. So, okay.
So great. Would you say that the coelacanth's survival till now as a species speaks more to some specific developed evolutionary mechanism or a different factor like the environment it inhabits? This is a wonderful, wonderful question. And George Gaylord Simpson, back in the 1940s and 50s, wrestled with this question, as have many paleontologists since. And the question is always, are lungfishes and coelacanths perfectly adapted to the habitats they live in, and that's why they still exist? Or do they just evolve slowly? And
here we have your attention, please. The Tompkins County Public Library closes in 20 minutes. Internet stations, Wi-Fi connections, reference desks, photocopiers, and self-checkout machines close in 10 minutes. The cash register closes in 5 minutes. To apply for a library card, please proceed immediately to the front desk. Thank you.
Well, I'll keep it short. So this is just an excellent question. And I really recommend, if you want to know more about this, that you look into questions about rates of evolution in different groups. I'm going to mention the loaded term living fossil because it needs to be mentioned if you're going to ask that question. I don't have an answer for you about living fossils, by the way.
Oh, come on, what's your answer?
I don't have one.
No.
I have a graduate student who's a specialist on that question, so I'll let him tell you. Yeah. Okay, so fin rays. This is really a great question too. Learned recently that fin rays are actually dermal bone. Do we know what they're homologous with scales? Yeah.
Ted talked about that a lot.
Yeah, good. Yeah, so the interesting thing is that chondrichthyan fishes, sharks, skates, and rays, and chimeras, have fin rays that are not associated with any bone because they don't make much, if any, bone. If they make any bone, it's associated with the basis of their placoid scales. But yes, in fact, fin rays are dermal bone around bundles of giant collagen fibers. So they're very interesting structures. And actually, the early lungfishes are a great important group to study if you want to understand much about the early evolution of fin rays. Okay, now this is a great question that I don't know how to answer. The question is, how is a plesiosaur's limb, which has so many bones, related to Hox genes? Good question. I don't really have any answer to it. But there's a guy named Lomax who's sort of the guy to talk to about that kind of question today. Dean Lomax, I think is his name, has a new book out on marine reptiles of the Mesozoic. And the last question here that I see is, why do coelacanths have low diversity but survive through that long time? That's another excellent question to which I do not have an answer. Is that related to how well they adapt to their environment? And that again goes back to the questions that were frustrating Gaylord Simpson before I was born. And actually, I will tell you, if you're interested in the history of paleontology at all, it's really worth going back and reading those books that George Gaylord Simpson wrote in the early 50s. Absolutely fascinating work. Okay, are there any other things that I should do?
I think we're good. So, Lauren, you want the mic and give a thank you to the online folks? Thank you so much.
Oh, no, I really enjoy being able to talk about this. Thank you.
So thank you to